Once again, welcome back to the session. So now we are going to discuss about the mechanisms or steps of DNA finger printing. So let me tell you one thing about here. Children, here you are not going to study in detail with regarding to the steps involved in DNA fingerprinting. Just we are going to name it, and that is what it's given in your textbook. So therefore, this is a 5 mark question, no doubt. They will ask you explain or list the steps involved in DNA fingerprinting. So it's your duty is only to list. But to make you to understand, we are going to explain each of this technique so that you can easily follow it. So now let me come for the steps involved in DNA fingerprinting. The technique involved is called as southern blot hybridization using radioactive labeled VNTR as probe, that is variable number tandem repeat as probe. One thing, what is this probe? See, this is small DNA fragments which have been synthesized artificially with known base sequences. I'll tell you why these probes are required. Okay, so with regarding to probe, when I come to a particular step where it is required, I'll be telling you about that. So now let me start with the steps. The steps included is the first one, isolation of DNA. But before going to isolation of DNA, the most important thing that is required is we have to collect the sample from the place of vicinity or from the vicinity of crime occurred. That is, I told you, this is one of the important steps used in forensic science department. What is forensic science department? Identifying the criminals. So to get DNA, we have to collect the sample. Let us assume there's a murder. So we people from forensic science department go there. They may get some what we can call samples, might be hair or skin or blood, whatever might be. So that will be collected from the spot where crime has occurred and it will be brought to the lab. So now from that sample we are going to isolate DNA. How this isolation is done? Might be the cells. Therefore, in the sample, whichever cell is present, the cell membrane will be dissolved, nuclear membrane will be dissolved, and DNA will be made to what we can call appear like precipitate like thread, and that will be collected. So that is what we can call isolation of DNA. That is, the DNA present in the sample will be taken out by what can say dissolving the cell membrane and nuclear membrane and by precipitating DNA. This is isolation. Second step is digestion of DNA by restriction endonucleases. See, DNA when we'll get a long thread. The entire thread may not be easy to what we can call trace out the sequences. So therefore, we have to break them into small fragments, and this is what we call digestion. So instead of digestion, you can use the word fragmentation of DNA by restriction endonucleases. What are these restriction endonucleases? These are a particular enzyme which are going to cut DNA at specific site called as palindromic sequence. That is, where in a DNA the nitrogenous bases will be repeated. About palindromic sequence you are going to study better in biotechnology. So therefore, by using restriction endonucleases, DNA sample will be cut into fragments. So this is what we can call digestion of DNA by restriction endonucleases. Third step, separation of DNA fragments by electrophoresis. You will be studying a technique called as gel electrophoresis. So here, now the DNA which have been broken down by restriction endonucleases, the length will not be same. So therefore, they have to be separated. So hence, by using a technique called as gel electrophoresis, DNA based on their molecular weight will be separated. So about this also you are going to study better in biotechnology when you study chapter 1 under biotechnology. The next, once you get DNA fragments, they will be transferred to a synthetic sheath of nylon or nitrocellulose. That is, in gel electrophoresis, you will be separating DNA fragment in the gel, which will become thick. Later on, those gel will be taken by using southern blotting technique. What is the southern blotting technique? The DNA fragment which is present in gel will be wrapped with what we can call blotting paper along with nylon sheath, and it will be pressed. When you press it, DNA from agarose gel will pass through blotting paper and come to nitrocellulose sheath or nylon sheath, and it appears like the band. So therefore this is what we can call it as separating or transferring DNA fragments from gel to nylon sheath or nitrocellulose sheath, and this is done. That's why the name, the technique involved is southern blot hybridization. That is, after transferring the DNA from agarose gel to nylon sheath, that technique is southern blot technique, and after that pairing will be done. Now the next step is hybridization using labeled VNTR probe. Yes, here one more thing we have to do is, you know that our DNA fragments will be having what we can call double helical model. So therefore there are two chains of DNA. Once you transfer it to nylon sheath, what they will do is, they will break hydrogen bonds by using again enzymes. So the two what we can call chain of the DNA will separate. Now they will remain as single chain. So now what happens? This single stranded DNA will be allowed to pair with VNTR probe. I told you what is while telling about variable number of tandem repeats, what is probe. So what are this VNTR? That is, this is the small fragment of DNA which is synthesized by man with known DNA. See, that is, we are going to synthesize using different nitrogenous base pairs which we know. So now these fragments will be laid on nylon sheath, and you know that the DNA of these what can say DNA in that collected sample will be single stranded. When you allow this VNTR probe, they will go and pair based on complementarity or based on complementary base pair. So that is what we can call hybridization, joining single stranded DNA from the sample with VNTR probe, which is known base sequences which has been synthesized by man. So hence you will and one more thing is, this VNTR is radio labeled. That is, we will have a nitrogenous base pair which has got a radioactive property. So why we make use of this is, when you use this and when you take photograph, it will appear like a band. So therefore, to get that band, we use radioactive VNTR as a probe. So therefore, by using VNTR probe, we are going to pair the single stranded DNA from the sample. So that is what we can call it as hybridization using VNTR probe. Later on, detection of hybridized DNA fragment by autoradiograph. What is this autoradiograph? Taking photography of whatever band you are going to get on nylon sheath. So when you take photograph, the original DNA sample which has been collected from what we can call vicinity, that will not show any any band, but the radioactive VNTR probe which has been taken, that will give a characteristic band. Since you know nitrogenous base pair on this band, you can try to tell what is the complementary base pair. So therefore, by using what can say the autoradiogram, which is obtained by photography of VNTR probe with the sample of DNA, where that will be the autoradiogram of the band will be of different sizes. Now, based on complementarity, you can try to identify the pattern of that particular DNA. So this pattern of a band is going to differ from one individual to another individual in population, even in a family. Only one place where this band will be same is monozygotic twins or identical twins. That is, if both the twins bonds are, okay, so usually of same sex, if they are developing from single zygote, in those children or in those individuals, this DNA fingerprinting may not work out very easily. But in others, this band is going to be different. So now, based on this band identification, we are going to, what can say, identify the DNA sequences. So this sensitivity of the technique is used, it is increased by using polymerase chain reaction. That is, what is this? Here, if you get, usually in the sample what you collect, it will be very less. Might be sometimes DNA obtained is very small amount. It may not be possible to identify to carry out this DNA fingerprinting. So therefore, before carrying out what can say gel electrophoresis, we are going to, what can say, make multiple copies of the DNA from that sample, and that is what we can call it as polymerase chain reaction. Once again, about this PCR, polymerase chain reaction, you are going to study in biotechnology. That is, after collecting the sample, the DNA in the sample will be multiplied in thousands or more than lakh, can say can say copies. So therefore, in case if we fail to identify in one sample, we can make use of the existing fragments of DNA and we can carry out this DNA fingerprinting. So now I am going to show you the photography of three important individuals and how DNA fingerprinting can be identified. So here you can see these are what we can call suspected people in a crime, or let us say in a dispute. So DNA from individual A is taken, DNA from individual B is taken. After carrying out all the DNA finger processing, you have got the radioactive band of three different chromosomes. You can see chromosome 7, chromosome 2, chromosome 16. So two individuals. Now after that, you have also got one more band, DNA from the crime scene. That is, from the sample which was collected at the place where crime has occurred. So that band also you can say, after DNA fingerprinting, you have got a band. Might be when you collect a sample, you may not know who is the culprit, but you will be having doubt. That is why from the doubt person, the blood is taken and from them DNA is extracted, and even in them DNA fingerprinting is done. So you can see top two bands of chromosome number 7, 2 and 16, the band can say DNA fingerprinting as obtained. Likewise, from the sample which has been taken from what we can call site of vicinity, the what we can call DNA fingerprint as obtained. Now you can see, compare. You see individual A and the band from crime scene. Chromosome number 7 will not match at all. Chromosome 2 might be, and chromosome 16 there is no match. So therefore, individual A is not a culprit. Now you see individual B. Chromosome 7 matches, chromosome 2 also matches, and the arrangement of nitrogenous base pairs in chromosome 16 also matches. Yes, here the person who has committed a crime is nothing but individual B. So therefore, like this, by comparing the DNA fingerprint from the sample and from the suspected person, you can try to identify who is the culprit. Okay, so this is with regarding to DNA fingerprinting. So children, please go through this. This diagram you need not practice. No one will ask you this diagram in exam. Only try to remember the steps of DNA fingerprinting. So this is with regarding to the procedure of DNA fingerprinting, and as a last part of this topic, we are going for applications of DNA fingerprinting. So let us see what are the uses of this DNA fingerprinting to mankind. So yes, DNA fingerprinting is the basis of paternity testing in case of disputes. As I told you, that is, Let us say a person in your family has been missing from a very young age. Later on, he will come after 20 years. Let us say he or she may come and tell, I am your brother or sister, or he may tell he is the child of your parent. Now, how to trust? Like that, many can come. So, therefore, if you try to carry out DNA fingerprinting of those, the suspected person, and also the DNA fingerprinting of your parent, if the matching is there, you can try to find out. If the DNA band does not match, then the person is telling lie. If it matches, then you have to agree. So therefore, in identify, that is, even in government hospitals or in hospitals after delivery, there are missing case of children. How to identify that? That is how DNA fingerprinting is going to help us. Second one is, it has application in forensic science. That is to identify culprit, crime. Third thing is, it has much wider applications such as determining population and genetic diversity. That is, for example, in human population, I told you there are races. The characteristic feature of Indian people differs from the characteristic feature of Europeans. So why this variation is there? If you want to understand, you can go for using DNA fingerprinting. Likewise, the diversity, I told you what is that, European, Negro, Chinese, Mongolian, Indian. So there are variation in the characteristic feature of this human races. So why this variation has occurred? There should be somewhere changes in the arrangement of nitrogenous base sequences among the individuals of this people. So how to identify that? That is by using DNA fingerprinting. Even to study to carry out population census among wild animals, this DNA fingerprinting can also be used up. So this is with regarding to DNA fingerprinting. So with this, I'm going to end up this session. Now, in the next session, I'm going to give you assignment. Children, please try to answer this, and very shortly, you will be given the information by college authorities that you are going to submit your assignment book you are going to submit your assignment book to Little Sai Angels, where that books will be given to us. So please keep it ready, and they may give you some dates so that you have to complete and submit it. So we will meet in the next session with assignment questions for molecular basis of inheritance, and for what is that, human genome project and DNA fingerprinting. So until then, good.